the negative lenses. And you then follow essentially the same idea, just like you don't go right after the lens, but you essentially go left after the lens after going through the center point, and then the parallel and the infinity ray, and then you form the image on the other side. So it's then a virtual image, which is not really existing because it's not on the right-hand side of the lens, but it's a virtual lens on the left-hand side. And so when you then know, okay, I have these rules, and I use my lens, just a very simple lens, for example. I have one here, it has 50 millimeter, and there's actually also a very simple trick to measure the focal length. So you point that towards the sun, because the sun is almost infinitely far away, and we've learned something that's infinitely far away. It produces rays that are parallel to each other, and then they focus in the focal point. So that's the definition of the focal point. So if you want to know the focus length of your lens, just point that to the sun, because it's almost infinitely far away, and then measure the distance where you have the small spot size on, for example, your hand or your screen, and that's the focal point. And then when you know that there's something in the focus point, it's brought to infinity or propagated to infinity. And then your eye is essentially then bringing something from infinity again back to the retina. And then the ratio between the two lenses here make already a magnification. So you have a magnifying glass already made up by two different lenses, or like one lens and your eye lens in combination. And then by varying the position between the object and the lens, you can actually change the magnification. So you can try it out if you have a simple lens, and then try to focus, and while moving the lens, you will see that there's a magnification effect there. And then you can use it. So for example, in binoculars or telescopes, you have two different lenses where you then have, again, the ratio between the two producing a magnification. So an object that's far away, for example, a tree or a ship or a boat or something, is then focused on one point, and the other lens is essentially then just re-imaging the focus point here. And then you see that the diameter of the two beam sections have they're varying, and that's depending on the focal lengths of the two lenses here. And so the magnification is basically the ratio between the two lenses here. Y minus, because the image flips, so it goes here through the point, and then of course the rays here end up here. And that's also what's happening in microscopy. So so if you have a point here, a small point, then you magnify, end up with a big point, and then again the ratio is the same. It's just if you compare the two, it's just different from the point of view. From here, it's like you have infinity to infinity, because then you watch with your eye, and from here you have an object to an image. So then you have no lens afterwards. So, and then we have, I think, also learned some aspects about the interference, which is then essentially, I think here I should maybe start. So when you have different waves interfering one another, so you see these Moiré fringes, for example, or here what we've seen in the Michelson interferometer, you have the resulting wave that is then essentially the sum of the two waves here. And why is that interesting? So we usually see light also, besides the ray character, as waves. And so when you have the wave here, similar to water waves, and you have something in the way, an obstacle, for example, and you have then the very tiny slit where only at the slit you can see the waves going through, then you essentially, the waves, they don't have any chance to go through the border here, so they only can start emitting new waves, so-called spherical waves at that point here. Okay, that's cool. And now if we go one step further, we don't have only one slit, but many slits. So in optical terms, we call that grating. These spherical waves are then formed at all of these little spots here. And what you see here now is the same thing what we've seen in the very simple example, where you then have two waves superposing, but now you have many different waves. And when you see here, there's something being formed. So you have, some brighter spot here in the center, and then one brighter line here and one brighter line here. And so this all boils down that you have a grating, and you have a single frequency waves, and then the light wave is essentially diffracting at the grating here. And so what you see here is the diffraction pattern. So this is nothing less than the superposition of many of these different waves here. So these become spherical waves. And then the further you go out in the infinity part, so just imagine you go very, very far to the right-hand side here, so infinity essentially, then these beams become essentially discrete. So you then have 3 discrete points, or in fact, you have many discrete points. And so this is how images
क्या
are
पढ़ रहा है
being,
भाई?
because you have not only a grating of one frequency, because it's 1, 0, 1, 0, 1, 0 at a certain spacing here, but it's usually a sample is made of many, many different spacings between, for example, proteins and well, essentially atoms. And so they form this optical grating, which is very, very complex. And what we try to do with microscopy or in optics in general is try to collect as much as we can from the light here and try to reconstruct, or like try to get an image of what is causing this diffraction pattern. And what's a bit nasty, unfortunately, is, so you've seen that there's an angle. So let's just go through it again. So the light here, for example, our illumination, our laser, or our LED is illuminating our sample, and then the light is here being diffracted, and there's a certain angle, and the angle is depending on the wavelength we have for illuminating our sample, then the spacing of the different elements here, and then essentially that's about it. So the smaller the wavelength we use, the smaller the angle becomes, and the smaller the grating becomes, the larger the angle becomes. So, and now imagine you have,
क्या पढ़ रहा है अभी? क्या नहीं तेरा तो इससे?
so sorry, somebody needs to turn down the microphone. Sorry. Right. And so
वो हुडा गैंग के नई मेंबर है जो तुम समझ नहीं गैस नहीं कर पा रहे थे ना, वाइल्ड गैस
I think
टी
somebody
207 में रहती है वो सारा दिन अभी था छावड़ा.
can you turn down the microphone eventually remotely? Okay, anyway, right. And so the problem then comes that you at one point cannot collect the diffraction orders anymore. And then you become, well, then the microscope becomes a problem, well, diffraction resolved, so you cannot resolve any better. So this formula here, what I just mentioned, is like the wavelength is the lambda here. The M is the grating order. Let's say we want to go for the first one, so we can neglect that. And the grating G is essentially the distance between the different spots where the light can enter through. And so at one point, depending on the spacing of the grating here, the angle become larger. And then at one point, you cannot collect the diffraction orders anymore, and then your optical system cannot transmit any information anymore, and then you lose resolution. I think to make it a bit more clear, there's a very simple analogy where we can see that every day. So if you somewhere where it's dark right now, you look in the sky and see many of these little dots, stars, then you may have the impression that some of the stars are bigger than others. That's actually only partially true. It might be true, but our eye is also diffraction limited. And since these stars are so small, the optical or the frequency spectrum that is emitted by the stars is so broad that our eye can only collect a certain limit of it. And then every star has the same width because our eye can only resolve them to a certain amount. And so this is essentially also the problem in microscopy, because at one point you can just not resolve any better. Okay. I'm just puzzled now by the yes, and so I think that's also where we stopped last time. So this is then also the formula for microscopy essentially. Okay, there's another thing which I wanted to explain briefly, that's polarization. So we learned that light is essentially an electromagnetic wave, and the electromagnetic wave can have a certain direction. So it can move, for example, along this direction or along this direction, and you can measure that. So for example, light that is emitted by a light bulb, like an LED or like a tungsten light bulb, is fully unpolarized, so every direction is equally available. Lasers, sometimes they are fully polarized, so they really just move along in a certain direction. And so if you have an unpolarized light source, for example, an LED, then you can make it linearly polarized, for example, with a polarizer. So then means like unpolarized light becomes linearly polarized. And then you can, again, take a second one, for example, to then eventually block it. So imagine you have one of the linear polarizers, and then you place the second one 90 degrees rotated, then there's no light because here it's unpolarized, here it's linearly polarized in that direction. And then if you rotate the polarization again, there's no light coming through. However, there's a funny effect. It's actually based on some quantum effects that if you place a third one, there might be there's still light coming through, which probably doesn't make sense. So it's really fun to read into that. So two polarizers mean there's no light, and if you have a third one that's coming afterwards, you have you have light because there's 25% of the linearly polarized light that's going in that direction still going through. And then where you also have these effects, for example, your screen from the laptops, usually they have LCDs. So you use the effect of linearly polarized light, and then you can rotate the rotation angle in order to kind of use it as a light valve. So there's either going more light through your pixel or less, and you essentially do that by rotating the polarization of the light. And what's also really nice is you can use that in microscopy. So polarization microscopy is one of these techniques where you can create contrast. And so it works like that. So you have a light source, for example, LED or tungsten or halogen, that is completely unpolarized. And then you have a polarizer right here. And then you have a specimen which can eventually rotate the polarization state. So for example, if you have birefringent or just some crystals, for example, if you have if you remember the birefringent or like the stress test of transparent plastic, so you take one polarizer, and then you have plastics in between that's transparent, and then the analyzer or like the second polarizer, then you can see, depending on how much stress the plastic is sensing, the change in polarization. And so since every different rotation of the polarization is then again interfering after the analyzer on the retina or on the camera, you can then sense that by essentially white light interference. And that's also what you have here. So this is essentially a crystal. And when it was crystallizing, there's some forces being formed here. And then you can see that at the further it goes out, there are some changes in the polarization. Which is really nice because there's a very nice application which I've seen recently. I think it's very worth looking into that because it's a really low cost and I think very effective way of detecting malaria. And what they do is here, they detect it using a cell phone. That's already pretty cool because it's low cost. You just have a little adapter which mimics a microscope here. But what they do is they essentially replicate this idea of having a polarization microscope. So you have an LED, again, you have incoherent unpolarized light, you have a polarizer, you have the sample, and then you have the analyzer, and then you measure. And what turns out, if you have your plasmodia, I'm definitely not an expert there, but I think there are some maybe in this meeting here. The hemozoin, which is I'm not sure whether it's expressed, but it's part of the plasmodia, which is the parasite that's causing malaria. That is also changing the polarization angle. So that means if you have this crossed polarizer microscope, you can sense the hemozoin, and then you can essentially detect malaria pretty in a very sensitive way. So, and it's still very low cost because you only need two polarizers and then you just cross them, and that's pretty much it. Yes, and I'm quickly checking. So, okay, so then maybe also looking in the just loading that one. It's the real one. Sorry. Right. Okay, sorry, a bit unordered. Okay, so because this bridge was missing anyway. Coming back to the resolution thing. So I mentioned our eye is also diffraction limited. We cannot resolve any better than basically what our eye can sense. Same is holding true for microscopy. So these are many, many different viruses, all labeled with some fluorophores. And it can be that certain are smaller than others. Maybe here there are even 2, 3, but we cannot detect it because below 200 nanometers, it's really hard to resolve that. However, there are some methods where you, for example, apply super resolution. One of them is called D-Storm, where you then label your sample with some fluorophores, and then you actually apply certain chemical protocol where the sample is not fluorescent all at once, but spontaneously over time. And what you then see here is many of these little spots, they are all diffraction limited, so you cannot resolve them better, but you know where they are from, and then over time you draw the picture as nicely depicted here in the demo here. Right, so you may ask yourself, okay, what's fluorescence? Fluorescence is just one more way to give a contrast to your sample. So in microscopy, it's always very important that you can see something. So imagine you have a white egg on a white piece of paper and you just illuminate it with white light. You will probably not see a lot. So if you have a polarization microscope and a sample that has some polarization features like microplastics, for example, because it has stress and then you can visualize that, for example, that's already good. Also, if you maybe just draw a black line on your cover slip, then you also see that, of course, because it's absorbing. That's what we learned last time. But sometimes you don't have a lot of contrast. For example, human cells, they are almost transparent. You cannot see them. And also, you have many different compartments in the cell, like the nuclei, maybe the cytoplasma, and then maybe some mitochondria. And this whole field of fluorescent microscopy essentially tries to use fluorescence where you have a certain molecule that is absorbing light. Then you have an electron that's going from the singlet state into an excited state. It's from the ground state to the excited state. And then essentially what's happening, there's some energy being transferred to the sample, to the surrounding. And so the electron is, well, reducing its energy levels slightly. And then when it's relaxing, which is usually very, very fast, it's emitting a photon. And what you see here is that the difference in the energy is essentially causing a slight shift in the wavelengths. So you, for example, illuminate with blue or UV, which is very low wavelength sorry, it's very low in the wavelength spectrum. And then you emit a photon that's towards the green. So you can then just apply a filter set where you then block all the blue light, but let the green light through. And then you have the ability, for example, to, well, express GFP, which is the green fluorescent protein that's then maybe just labeling the mitochondria, for example, or something. And then you can specifically target different cells and then create a contrast, which is really nice. There's also synthetic dyes where you then, for example, use antibodies to label the actin filaments, for example. So, and then you can apply all the different dyes all at once and have a very fancy microscope to basically probe and produce contrast there. Right. So this is one laser, and many dyes are sensitive in the UV, so you can excite them in the UV. So if you have a UV laser, then many of the dyes will just glow. Okay. Let me see. Okay. Let me just, let's go. So some optical applications. I'm not sure whether I went through this one. What we try is using these here instead of those. So be frugal. I think we went through this already with the optical pickup units that you can use them for microscopy. Well, there's obviously a very long history about microscopy. Right, so the single lens cameras also there. Okay, I think this one we haven't talked about. So this is that's something we have worked on, I think, for the last 2 years. The preprint is, well, almost published. We're still waiting until it's fully there. Nevertheless, this is all open source. So you can find the sources, either you Google espressoscope or better Matchbox scope, because that's what it was called before. And it has this ESP32 camera where you can just use it for different applications. So that's essentially what it boils down. And so this well, it's a typical microscope. You have a little lens that is essentially just in macro mode. You can use the internal LED for illuminating the stuff, but you can also do more with it. So for example, if you add a grating and then a little slit, for example, you can just repurpose something from the 3D printer, which is a nozzle. It is a perfect small diameter. You can get it down to 0.1 millimeter. It's typically very high quality. So that's something you cannot print. And so then you have the entrance slit. And then you have the grating, and then what's happening, the grating is basically diffracting. That's what we just learned. And then you can print or plot the spectrum here. And so that's what we also did. So there are all these building instructions online. I can show you how that works in a second. And what you see here is essentially just the picture on the camera. So you would still need to calibrate the pixel numbers to the spectrum, because you wouldn't know whether this is now 450 nanometers or 600, but you may have a rough guess because LEDs usually have a certain spectrum, and then you can calibrate with that. What you can also do is, since it's so cheap, you can just put that into the water and, for example, observe biofilm growth. So this is just put in a watertight container. And then you have a little lamp, a battery, and then it's just observing what's happening over many, many hours, if not days. And what's really interesting is, well, this is a time-lapse series that's going for, I think, 2 days, that there are all kinds of little creatures are coming. So I think the resolution is not very good. It's also relatively blurry because underwater there's all kinds of parasite light. But if you just go forward a little bit, there are all these little things where we still don't know what it is. But since it's captured on a cover slip, once we have that, we can just put that on a bigger, better microscope, and then we have the correlation between the time and then also the the optical resolution to see what this actually is. And the good thing about this is it's really low cost. So if you want to replicate that, it's fairly easy, I'd say. Yeah, so it's maybe a bit boring to just observe the whole thing. Yeah, one other thing, what we're trying to do here is observing the sleep behavior of other model organisms. That's called nematostellas. It's just one application where you, for example, can use Frugal tools. And so what we did essentially is we have many of these different or many of these small microcontrollers, and then put them in an array, and then every camera or every microscope is then literally observing one of the wells or one of the animals. And so like these Frugal tools allow you to do things that were not possible beforehand, because this is if you just count the parts and 3D printed parts, it's relatively low cost. I think it's somewhere around 300 euro. And I think this whole maker movement where the big manufacturers are pushing to make things more available to the people so that they, because they see a market, it's very beneficial also for science, because then you can repurpose things for science, essentially. So here we have these little cameras that's borrowed from the espressoscope. Just to show you, they get relatively warm. That's not so good for animals. And so the basic prototyping thing was we had these banks of the little cameras, put them together, edit the lenses to form the little microscopes, and then you have the full microscope. And also this whole thing, of course, is open source. So it's still in the making. It's not fully done yet because we're not so lucky with making all the cameras working all at once. It's a challenge on its own. Yeah, but you can also use those low-cost things, well, of course, for teaching. So that's something we did last year in Nigeria, but also for something a bit more productive. I think Manu is now in Senegal for giving people a course on how to identify malaria. It's a huge topic, and I'm by far not an expert, so I've just touched this thing barely. But the people in Nigeria, they really rely on these optical tools to detect malaria, and this is also based on this low-cost camera. So that's something that people built on site, and so it's really difficult to see something here. So this is a staining, a Ginza staining. That's essentially just labeling the DNA, if I'm not mistaken. parasites. And so these little spots here, that's something you're looking for. So I think here we are really at the edge of this €5 camera plus a €10 lens. But in theory, it's possible. And also you can teach the people on the ground how to build these tools and also to repair them, because everything can be printed, which I think is really nice. Then, of course, one thing, then you think, okay, you have a microscope, you have the optics ready, but in malaria diagnostics, it's really, really important that you have, or that you see many, many cells so that you can get the statistics, how many cells are essentially infected. And so this is maybe just a little tour on how to think, for example, microscopy differently. Because, I mean, if you want to have something that's replicated, in best case, in the hundreds, it should be really low cost, so not cost much. And then, but still you want to scan your sample. And so this was one idea we had, which is essentially similar to a vinyl player. So you have a little motor, but it still scans in XY. So it's moving from the center outwards. So if you have a sample and the lens mounted on top, it's essentially doing a spiral motion so that you see, when you're fully done with the whole spiral motion, the full sample. And cost you almost nothing, because these motors, they are like $5 or something. And then the parts on top, they can even be less. That's all nice, but mechanically, that's well, always a challenge. Well, maybe this is the next iteration where we also had like a light source that's then driven with, again, maker-based equipment. So this is a RFID-based technology, let's say, where you use induction for getting energy through the air. Not very efficient, but it works. So then here you would see the lens coming from above, and then we do the imaging of this malaria, like the blood smear here. That's all very nice, but I think we still need to improve a little bit, because mechanical scanning was challenging, because the slide always had a little tilt, so we could not really go for that. So the next iteration, and that's also work in progress, so it's not published or anything, but just to give out some thoughts what's out there. So I think maglev, I think it's not a big thing in the US, at least. Here in Germany, it was for a short period of time, and I think now it's not anymore. Nevertheless, it works by having a magnetic field that's essentially alternating, and then maybe you have a constant magnetic field. And then what's happening is that the magnetic fields are essentially just causing motion. So that's what you see here. So if you alternate the current through the different lines here, that means the car here is moving back and forth. And that's all very nice if you want to move a sample in one direction. Apparently, so the first test here kind of works. And what's nice about this thing here is you can just order this PCB here, which is literally just a 4-layer PCB with all the electronics on board already. And then it's perfectly plain. So there's no focus variation, because anything is already in focus. And what's cool is, if you just think, okay, I have this magnetic field that's alternating in one direction, why not just use two sandwich layers of these here crossed, so that you have the X and Y all in one PCB. And that's, I hope, the next slide here. It's not really fancy, and it's not fully applied to microscopy yet, but that's the next step here. So yeah, so this here is then the motion in X and Y combined. So the hope is now a bit, together with the optical feedback, to have a controlled XY scanning in a microscope that's essentially created by these little cubes with UC2, with a little Raspberry Pi camera, and then put away all the load of building an XY stage, and then also, well, paying for that, because usually it's very expensive, to have that in a kind of solid state device, which costs almost nothing. So this is something that's very nice, because you can use a lot of the tools from the maker communities here. Okay, maybe it's two more of these weird things. There's another guy in the Netherlands, Vittorio. He's also very keen on making things for the scientific community, especially open source. So this here you find on printables, that's something similar to Thingiverse, for example, just a repository for printable files. And this is a very nice thing, because it's so simple. You just have a small well, it's called bike particle accelerator. And so you have a little wind tunnel, and at the end of the tunnel, you have this little slide that you use for collecting stuff that's in the air. And so what you do for here is just do measurement of the air quality while using the bike. And we brought it a little further because we wanted to correlate the position and also the time, or like in best case also the location. And for this we used a cassette. So then we just, I don't know whether you still know that, back then people used audio cassettes for listening to music, like with a Walkman. And so you have tons of these cassettes available. So this is one from my parents. And so what you can do then, so the pen here has some acetone with some nail polish. And so then you coat the tape essentially, and then you essentially collect the particles and they are getting glued on the record. And then when you're back home, you can just put it under the microscope, and then you essentially can detect, for example, microplastics, what you've collected on the bike here. So it's not perfect quality things, but yeah, also here, open source, if you think that's a nice thing, it's all there. Right. So these other tools, I maybe just briefly mention why sometimes I think it's a trendy thing to have a bigger, better microscope. So better camera, better stage, everything better. So that's also why people always apply for these big grants. But sometimes you don't actually need to do that. So you can, for example, repurpose an old microscope and still do cutting edge science, which I mean, it's probably not necessary to do that here in Germany. Because I think Germany is a very well-funded country, but nevertheless, personally, I'm very curious whether it could work. And I think it's also nice to show that you don't need to apply for a million grant to just show that something works. So here, for example, thinking back about the super resolution methods where you have different items that are blinking, and then you basically conclude their position. So you have fluorescence, and you change the protocol slightly so that you still excite an electron using your laser source, then it's emitting light, and then you have the shift. So you then have the different filaments here all being excited and then emitting light collected by the microscope. So your sample would maybe look like this here, very blurry. But then there's a technique, it's called storm, as I just said, where you then increase the laser intensity and also change the buffer. And then what's happening, the electron is not being relaxed in the very same moment, but a little bit later. And so then it's kind of trapped in a dark state. And then it's relaxing after a certain time, which may be up to minutes. So then it's really just staying there and idling. And then at one point, it's relaxing some hundreds of photons, and then you can collect them. And so this is what's happening here. So all of these blinking spots here, they just emit a photon, or like several photons, and then you can detect, okay, what's the origin? From this knowledge, you can then localize each of these different molecules. And then when you know the position of all the molecules across thousands of frames, then you are in the super resolution world. And this is then collected or like acquired with a cell phone. So we literally replace the 20,000 euro camera with a 200 euro phone. Fair enough to say that it was a monochrome camera, so it had the ability to collect more light because it's not blocked by the color filters, as you learned it last time with a Bayer pattern. Nevertheless, we got down to 80 nanometer. And if you then, so yeah, and how that usually works, because, I'm jumping between different slides now. We wanted to apply that to virus imaging. And we know viruses, that was a big thing just 2, 3, 4 years ago with SARS-CoV, and they're really small. So the SARS-CoV or the SARS-CoV-2 virus, the virus itself is roughly 150 or 100 nanometer small. And what we've learned is that the optical resolution limit is somewhere around 200 nanometers. So we could only identify the spot. So we could not identify any of the receptors or anything, or the spikes or so. So the way you can do it is you label certain compartments. So for example, the spike, there's an antibody that's labeling the spike, that's typically very, very small. So this is not the right scale here. So antibodies are a lot smaller than the viruses. Nevertheless, they have a size. And then you have a primary that's then labeled against a goat, for example. And then you have a secondary antibody that has fluorophore. And then if you have that, then you can label your viruses, and you can specifically say, because the antibody binding is so specific that everything that's fluorescent is a virus. And then just going through. So this whole thing is sometimes called ELISA or just antibody staining or immunostaining. And then what we had here is this little microscope, again, based on the cell phone, and then some optics here. So we essentially also moved away from the big Zeiss stand, and then you could do this measurement even on the cell phone. So we trained a neural network and tried to reconstruct also the frames on the cell phone. And it's really impressive what cell phones can do. However, the problem is a bit because, I mean, of course, we have to push down the price, but at the same time, we should not forget about the reproducibility. So that's one experiment that we do here is maybe also reproducible and have similar results somewhere else. And the problem with smartphones is that with every firmware update, maybe a function from, for example, accessing the camera is removed. And so we even had the problem to access the monochrome camera. So we had to hack into that. So it worked, but it's not really reproducible. And that's a bit the next story, what I wanted to talk about in terms of reproducibility by means of modularity. And so my background is very much away from academia. So I did an apprenticeship as an electrician at one point, and I did, well, usually what you do when you work in electronics is developing circuit boards, and it takes a long time to actually build one of those. Programming is basically never working because there are so many errors. And so when I was done, there was this little microcontroller, and maybe a few of you have seen it already. And it's an Arduino. It's an open source microcontroller that you essentially just connect to your USB. It starts blinking automatically, so nothing to do here. And what's really cool about it is that it has an open source community. So that means you can get code from people who already worked on that, for example, in home automation or robotics. And then you can just really quickly do rapid prototyping on all kinds of different projects. And we wanted to do the same thing for optics, essentially. So you have the microscope. I think what we have learned is that you have several lenses, and they all interact by concatenating them with their focal lengths. So you have a light source.
Benedict?
Yes?
Sorry, we just have a quick question.
Sure. Sorry, yeah.
Yeah, sorry.
I just had a question about your Cellstorm 2. So it's like achieving like similarly like super resolution, but how thick of a tissue can you image? Can you go actually?
Oh, sorry. Yeah, my question was basically if you can image a thicker session with Cellstorm 2 and potentially maybe even creating like a cell chamber to allow some live cells and whatnot to be imaged.
Yes and no. So I was skipping the Cellstorm 2 bit a bit because I didn't want to go too much into the details, but just briefly showing the principle behind it and maybe then answering your question. So Cellstorm 2 is based on a system that is not a typical wide field microscope, as you see that here, where you have a laser illuminating the full sample. It's based on turf. So like something we learned last time with the waveguides, where you then couple light and then you have the total internal reflection. So that's what turf stands for, total internal reflection microscopy or fluorescent microscopy. And so what you do here is you have a photonic chip that is being fabricated, for example, now by this company, but I think there are many others, that is insanely thin. So you have 150 nanometer high silicon nitride on a substrate that's, for example, silicon dioxide. And then what's happening if you couple light into that, since it's an optical clear waveguide, so to say, is that you have light transported through this little chamber or chip here, or channel, it's more better. And then this evanescent field, what we were talking about last time, this field that's kind of lurking outside the waveguide is eventually being absorbed by the fluorophores and then also excites the fluorophores. So that means you see like everything in the turf region, which is, well, maybe another 150 nanometer can be excited. So that like everything else is dark in best case. I mean, there's lots of scattering light, but in best case, you only see the 150 nanometer on the surface of the chip, similar to turf microscopy. So thick sections usually don't work because if you, for example, have a nuclei here, it's a very big compartment of the cells, sometimes several tens of micrometers in diameter. Then you like all the different photons, they have to travel through these big compartments, and they scatter a lot. So then that's degrading your optical resolution. However, with the typical storm, I don't know whether I have the slides in here, like the storm that we used on the UC2, which I'm showing in a bit, also open source, that can do thick sections by means of you're not bound by the chip's surface here. So for example, here, these bacteria here, they're sitting on the chip's surface. You then have this fluctuation of the intensity, and then from the fluctuation of the intensity, you can then also compute a better resolved image, which should show here. Doesn't, okay. Can do lifestyle imaging with the cell phone, but you see the resolution is not really good then. So that was a long time series, and I think the cells also didn't survive that much. Yeah, here are the bacteria, and these are then the membranes you can then reconstruct. So so long story short, like thick samples or like even tissues with this chip approach, that's not really possible. For volumetric imaging, we have different tools, which I may also present in a second. Does that answer your question already?
Yes, I was just trying to see if there is a volumetric imaging for like light sheet microscopy, but Frugal.
Yes, yeah, this is coming. I hope I have the time in a few more 100 slides. Now I think I'll come to the point with the light sheet microscopy and how to do that frugally. Right, and so just like from the microscope point of view, so you have an illumination. The illumination is being collimated. So this typical thing is called Köhler illumination. You then illuminate your sample with nicely shaped light because it's all parallel, then homogeneous in best case. And then you have your microscope objective lens. It's then being imaged, or like, yeah. And then a second lens, usually called tube lens, is then forming an image on the camera. So this usually is called Fourier optics or infinity corrected optics or 4F systems or 6F systems. However, what's important is that these different focal lengths are snapping into one another, so that then essentially the focal length of this lens follows the one of that one, and so on and so forth. So you can just modularize your system already, and what we essentially did with UC2 is just building little boxes around it. It calls it UC2. That's it. And there's a story behind it. So we did not start with cubes in the first place. So it was a long iterative process where you, well, there was somebody actually wanting a small system where you can image cells in incubators. So that was the first task. And then he wanted to have focusing, and then he wanted to have fluorescence, and then. And so then what came out was that we wanted to build one cube for every of these ideas or modules, so that every cube, for example, is holding a lens or maybe a laser or maybe an eyepiece, and then you can construct different setups relatively easily, which I'm trying to do also in a second. So, and now we're even at the point where the 3D printed parts, like the cubes here, they're not printed anymore, but injection molded, so that you have better reproducibility, so that and also it's much faster to actually fabricate them. So in this library of different parts is online, so you can just go online and then download the parts. So maybe just quickly checking how that looks like. You go to the UC2 configurator, then you, for example, select the incubator microscope, and then it gives you all the different parts that you are downloading them. And this zip file then has a certain amount of these puzzle plates, and then you can open your printing tool, and then essentially just drag and drop all the different parts, and then prepare them for printing. I hope you can see that. So this is then the mechanism how you would do that. So this is, well, for this you would need 2 print sessions on a Prusa, and then you just, well, if you slice it, then you see how this would go. So maybe, I don't know, 16 hours or something. Oh, well, 7 hours. And then this is what the printer would do then, like many, well, this is not 100% infill. But then in the end, you can replicate this system, for example. And you can also create your own modules. So if you go to our GitHub web page, I think if I'm not mistaken, if you go to template, I think that one. Then we give you all the necessary tools to know, okay, so because some people use Autodesk Inventor or Tinkercad or SolidWorks or you name it. And so what you have here are the was the standard what we once put up there so that you know, okay, this thing what goes into the cube here, which for example holds your lens here. Well, here there's none of them in the printing area. Well, this is a very funky piece here. But there are more here, for example, for the camera here. That means like, so if you stick to these dimensions and you print them out, then you're compatible with the system. And that was a bit the idea, how to, why we came up with this thing, because there are so many different optical elements like mirrors and lenses and objective lenses and actuators. It would be nice to have a common standard, similar to, I think, what Thorlabs is doing. It's more of the professional side. But more in a, well, let's say frugal do-it-yourself side. Yeah, so I invite you, maybe I just share the link also in the chat here, just in case somebody wants to look at it, and continues the slides here to maybe give some more ideas what you can do, because it's modular in the sense that you can essentially add whatever you like. So you have the inserts for adding mirrors or lenses, but you can also add external parts like electronics, have bigger compartments, like, for example, like little projectors that are then being used for projecting patterns into the sample plane, or yeah, and then all like if the mix or the combination of the different parts then make a system. So this here, for example, is a light sheet system that you can then construct from available components essentially. And so maybe just stopping sharing for a second and showing you how that looks like in reality. So I think we have the time for that. Okay, so I've brought some of these boxes here. So these are the boxes, and you have several cubes. So I can just present them quickly and maybe stop the time how fast I can build a microscope. So this is the essential part. It's an eyepiece. So then we have these cubes here with mirrors. And so what you do here is kind of a periscope. So you see the chat now. So this is light coming from here, then being redirected going up here. And then what we want to do is now forming a smartphone microscope. So what we do is well, maybe going online again, because there's actually also a documentation for that one. So there's another one, maybe a bit weird, but the documentation that looks a bit nicer is on openinstitute.jitter.io. Just sharing that link quickly. So for those who are interested, chat here. And then under educational kits, Discovery Core, and then English, then you see either well, some videos and like the way it's being constructed, but the more updated version is in the PDF. And then you see the explanation again of the different tools, like what is the lens, what is the mirror, and how they belong together. And then if you go through the whole concept of, like, for example, forming a telescope, well, some of the parts are not translated properly, I see. Anyway, you end up with the explanation on how to construct a microscope. So this is our microscope. We'll modify it slightly, or maybe not. So so this is what we do. So we have two of these mirrors. We have a Z stage, we have the objective lens and the light source and the sample and the eyepiece here. And on top here, we put our cell phone. So stopping screen sharing again and then just pointing to the camera. So this is what we have. So the manual actually looks a little bit different. That's totally fine. So we have this here now, two mirrors. What we are doing now is adding the Z stage. So this is a bigger component here. So this is then the stage here, and just adding more stuff here, maybe better to see that here. And the core idea is if you connect all the different cubes very tightly together, it's really stably. So the next bit would be the eyepiece. So that the eyepiece here, the optical path is now you look through this path here, it's being reflected here, and going through that here. And then we put our objective lens here. So we screw that in, it's an RMS thread here, and that's already a microscope. So the the final corrected lens here, I think it's further described in the manual, will form an image here, and then the eyepiece here will bring that roughly to infinity, so that if you look through this eyepiece here, you'll see what's in the focus of this lens. This is a bit impractical, and you cannot see that, so we'll do a trick. We just use our cell phone instead, so that we'll make the whole thing a little bit more stable by connecting all the different cubes tightly. And then we have a cell phone adapter. Well, something that's missing, of course, is the sample. So we have yet one more cube here for the sample. So it's just a magnetic clamp where you can place the sample on top of the cube here. And this is a random sample. I'm not sure what it is. Maybe somebody of you knows what it is. Martyr, Hunter, I don't know. Something I put here on the microscope. And so this is now a microscope. And well, certainly we are lacking the illumination, so we use this here and put that on top. And then we can simply use our cell phone camera. I'll show you in a second. Not strong enough. Okay. So, okay, and then what I'm trying to do, I don't know whether you can see that. I'll share. In the other camera. So, and here you see how easy it can be to construct a microscope. So you see already something. I think I cannot zoom in. Ah, yeah, you can. Interesting. Okay, so this is not in focus right now, so we will focus it. Yeah, the camera is also doing its focus on its own. So this is already the full microscope. So you see, it's really no magic. You just combine different cubes where you then have the light source. Here's the sample. So if I move it, you see that it's moving. Actually, quite funny. And you have a 4x objective lens. So it's not really high magnifying lens. That is then being moved up and down here. And then you have one mirror, the other mirror, and then the intermediate image, because the final corrected lens will form a mirror, an image at a certain distance after the lens. And then the ocular or the eyepiece is then transferring that to roughly infinity, so that the lens here from the cell phone can then image that on the internal sensor. And then it goes through zoom, and then now I see that on zoom, which is really funny. Okay. So this is how a very simple microscope can be constructed. And what you can do, for example, is you can then add different polarizers. So for example, this here is a polarizer, which itself is not so much fun. But if you have a second one and you rotate them at one point, you can fully block it. I don't know whether you see that. So I'm rotating one of the cubes, and both cubes have polarizers. And that's then the effect of you first polarize the light, and then you analyze it. And if the two are crossed, there's no chance that light will go through. So what you can do, I haven't tested it before, and it's probably possible. Let's see. So you can easily add polarization to the microscope. So let's try that. So we use the polarizer right behind the torch. So these cubes are made the way that they are point symmetric, so you can have these inserts just being rotated in all directions, sometimes handy. So now we have a polarizer. We use the unpolarized light, and then we put that here again for the sample. And then what we need to do is essentially adding the polarizer also to the after the sample, so that we kind of analyze the light. So for this, we use the second polarizer. You can rotate that. If you usually you call that analyzer, because then you analyze the state of the polarization. So we have here now the polarizer. So polarized light goes through the sample, and then the analyzer. Then we have to add the eyepiece. So everything gets one layer in between. And so what you're doing, maybe that is not working because I haven't tested that. It was a quick idea I had. So we use the cell phone again. Maybe zoom out. Okay, so let's see. Maybe not in focus. Okay. Bring that to the focus. Okay, so now we don't see anything. But what we can do, I think the polarizers are not good enough. There we go. So what's happening now is, well, there's lots of stray light. I think it's coming from somewhere, not sure. But now we created a very, very simple polarization microscope. I'm curious where the stray light is coming from. So when I'm rotating either the polarizer or analyzer, that means that the light is either going fully through the analyzer or is fully blocked. So either it's the two polarizers are like this or like that. So if the two polarizers are like that and there's no sample in between, we would expect well, a dark appearance. It depends a bit on the quality of the polarizers. They are pretty not so good quality. So this is the darkest we can get. It's not really dark, unfortunately. And then if we add a sample, the sample will then rotate the polarization state. So wherever you have this weird looking here, the bright spots here in the center, that's then caused by some compartments in the cell that are rotating the polarization state. So for example, the bright spot in the center, they are having some polarization properties that we can now identify with the microscope. Okay, so these are just some things applied that we may have learned. And with this, you can already, thinking back about the Imozoin, detect malaria with a better contrast. So going to the next thing. So this is all documented. And if you want to learn how to use it, there's, well, many things being documented here. And if you don't find what you need, just reach out. We will be very happy to help you. Okay, so we learned there are many different things that are inside the, well, you can put into the cubes. And then there's also something that's coming from the community. For example, the open flexure microscope. That's also a very impressive microscope. That's a single piece print that can do XYZ scanning. It's also compatible with our system because somebody made the adapter. Yeah, and then you can put that, for example, in the incubator, observe cells. That was the very first application where we wanted to use it for. You can add lasers, then you can, for example, observe fluorescently labeled bacteria in cells and put that in incubators. And you can also do storm on a typical microscope. So this is already relatively old data, so you can also again, go back here on the website and use the search engine and then add storm and then use the storm. And then you see a more in-depth documentation on how you would be able to build the storm system that's, I think, almost published in eLife with a relatively low effort. So if you have all the cubes, it's a matter of 2 minutes to put all that together. And then you can literally do high quality images here. So it's all very low cost. So I think roughly 2,000 euro with the camera and lasers and stuff. And so Ando did all the analysis, how stable it is even in incubators. And then also the imaging results to me are always very stunning. So it's yeah, somewhere around 50 to 80 nanometer, which I think for such a system is really impressive. Cool. And then, yeah, so this is all open source, just skipping that one. You can also use structural illumination microscopy, where Hauran spent lots of time on creating a computational microscope in a way. So you illuminate your sample with a structure. So, and then you use the Moiré effect to computationally reconstruct the blurriness in a way so that you get high resolution. And so I think that's something we learned last time already. So I think I may just skip that one eventually. So just to see how stable 3D printed things can be. So we learned lots of things about interferometry. So what you use here is a laser beam splitter, so that then the beam is split in two parts, and then you can observe the interference here, for example. And this the fringes. So every fringe is essentially just the constructive or destructive interference of the laser. There's also the system right here, but I think I'm just skipping that one. So this one, well, lots of source of inspiration. This is OCT, but I think this is a bit off topic now. Yeah. So then, of course, for microscopy, in the end, you also want to have a system that is then controlling all the different compartments, because what you have is many different actuators to move the sample around, maybe a laser or LED to illuminate your sample, then focusing mechanism, and maybe even something to keep ourselves motivated to survive, like a heating chamber. And so to make sure that all these different components are well orchestrated, we also use an open source software that's called ImSwitch, which we also co-develop now. It's all based on Python, so you can automate your experiments relatively easily with this one. Just in case you're curious where to start with, well, using all these components here. For example, what you then use microscopy for is large slide scanning. I think that's also what Honshwan and Manu and many other people in Manu's group are doing, essentially, is malaria diagnostics. And you have all these slides and you want to screen them very, very fast. And then there's, well, that's where automated microscopy is really, really powerful. And the tools you can get from Nikon, Zeiss, and all these other manufacturers, they're great, but they're also very expensive. So here, there's one device that's fully printed, and then you can use it for histological analysis, for example, and then have many of these tiles, stitch them together. And that's all possible by means of open source software and hardware, which is really nice, because then it's low cost and you can repair it on your own. It's not so difficult. And that's another thing which I mentioned. So this open flexure stage, that's from Richard Bowman's group in Glasgow. And you can also have a look. I didn't put the link here, but it's a really nice piece of microscopy where you literally just have a single piece that is printed like as one part. And then you already have a microscope, which very nicely can scan and focus your sample. Yeah. What's also nice is the software that I just showed with the ImSwitch is also, well, you can run it on Raspberry Pis, for example, so it's not super hungry in terms of resources. And then you could put it also, for example, in watertight containers, and then also operate that from remote. So that's also something that you find in plantoscopes, for example, which are really, really impressive. They're fully operated by Raspberry Pi, and essentially a flow stop microscope where you then have a sample from the pond or from the sea, like your plankton tow, and then you want to maximize the throughput by means of like, okay, image as much as possible as you can. Okay, so this is then also all nice, but I think there was also some interest in volumetric imaging, so skipping that for now. So why volumetric imaging is interesting? So when I was visiting Manu for some months, I think almost 2 years ago, that's a long time ago already, we were really interested in imaging plankton. And I think there are several reasons why to do that. One of them, they play a very important role in the carbon sequestration cycle, so that carbon is bound and then eventually going layers down, and then nobody really knows what's happening there. So I think it's a whole topic on its own. So I'm also not an expert there. But what's important is to know the shape of the different plankton or like diatoms and stuff like that. And so what you see here is it's a mathematical formula by somebody's art project where he came up with a model to render them. So they are not real, even though they look fantastically real. But what's important for us is like to get the 3D shape of the plankton. And so what you have in typical microscopy, well, we learned that there's a resolution limit in X, Y, but there's also a resolution limit in the Z direction. So I think if you ever captured any images from people, you always see the person in front and maybe also a blurry version of the people in the background. So this is because optics is always a projection along the optical axis on a single plane, because we don't have 3D cameras yet. In microscopy, there's a solution to that. Also in the real world, if you have 3D scanners, they work very similar. So you create a two-dimensional sheet, which is then illuminating your sample from the side, and then you're detecting only the part that are being illuminated with an objective lens that is being oriented perpendicular to the light sheet here. And then what you do is you scan the sample through this light sheet. And what you do is then similar to an MRI, have several slides, and then you can reconstruct the three-dimensional shape as a volumetric image. So, and how do you do that in Cube? Well, it's relatively straightforward. You, for example, have a laser that's either coming through a fiber or just out of a laser diode. You shape it with a cylindrical lens. Cylindrical lenses, what's shown here, it has only a single foci. So that means it's not a spherical shaped, it's just like a cylinder that's cut away. Then you form a focus only along one direction. So that's pretty much it. You then image that light sheet with a second lens, and then into the sample plane, you have an XYZ scanner so that you can move the sample. And then you have the microscope being formed by the objective lens, filter that's blocking the laser light, and then the tube lens and the camera. That's it. And then this is the very simple setup here. So similar to what you just saw in the slide earlier. And this is when you slice the 3D sample layer by layer. And so this is literally just GFP labeled zebrafish, where the neurons are expressing GFP. And then when you reconstruct these layers here, you get the 3D volume. And well, head is still missing. It was lost during the experimentation phase, not sure why. However, the problem is you have one fish, but if you have the whole ocean, it's really difficult to image all that. So what we wanted to do is lots of samples per minute, of course, so increasing the throughput. And so what we came up with is the following mechanism. So you have a flow-through microscope. So fluidics, usually they're very prone for high throughput. And then what you also did is rotating the sample. So so that means it makes, whilst moving through the focus of the microscope here, a spiral motion. So that's something you see here in the simulation. And so we wanted to measure two things. So autofluorescence of the sample. So many of the algae and plankton, they offer autofluorescence if you illuminate them in UV or some blue. And then also the scattering, so that we eventually have the three-dimensional scattering response so that we can reconstruct it eventually. And so we have holography and light sheet, and so that's why it's called holy sheet. And that's then how it looks like on real samples. So this is some I think algae. These are just some fluorescently labeled beads. And then while they are rotating, you can image them like slice by slice. And here you have then the projection, because we want to use inline holography. And then the computation comes into place where well, here the algae, you can relatively simple just say, okay, for every slice in the rotation, we just put that into a 3D volume. So it's basically polar to Cartesian transformation. So this looks like this here. So you then see the 3D shape. However, for the hologram, it was not so straightforward. So I think here we need to apply some more knowledge in order to get better images. But the overall system was really low cost. And just checking whether I have the slides here. No, okay, then maybe on another one. Here, probably. So you see, because what's really nice is you can prototype on one system and then deploy that on another system. And also here, I don't have that. Oh, shoot. Okay, then let's quickly have a look. So it should be open source. So it's either can search first here, and then should be either, yes, holy sheet. Maybe not the latest version, but some version. So this is then some version. Yeah, it's the standalone version, let's say. So this is then doing the same thing. You have a laser that's forming a light sheet, and then the light sheet is essentially here in that plane, and here you have a tube that's being rotated by the motor here. And with this, you can then also create this holy sheet on essentially any microscope, because it's having this multi-well format. So you can just put that on any microscope, and then you take 3D images. Good. Okay. So I think this is mostly what the last slides already. So just to show what's possible. So these are like just some random techniques I put out there. So lifestyle imaging, I think is relatively straightforward, not so expensive, 10K maybe. SIM, that's structured illumination microscopy, the one with the patterns, that's already a lot more expensive, up to 300 or even more. STED, that's something where Nobel Prize winners spent lots of hours creating them. It's really cool with donuts and stuff like that. So you can go down to 10 nanometers, which is very cool. And then this cost you 500,000 already. And the MinFlux, that's insane. It's like almost 1 million, but it gets down to 2 nanometers. And so what we have as tools at our hands now is it's a lot cheaper. But of course, you pay with the price that you have to invest more. So you have to create, you have to debug and stuff like that. And I think with this toolset, you're already very well equipped with imaging big problems, like especially with the storm. That's very nice. And I mean, the most powerful tools are mostly bright field anyway, for just seeing whether cells are alive or not. And so you don't need to spend tons of thousands of images, dollars to get a good system. You can just build it on your own. And make sure that it's open, because then it's reproducible if you can share the sources. And if you don't find that in your PowerPoint presentation, you better have that somewhere in the cloud, so that if somebody wants it, you can just download it. And so how do we do that? So we use GitHub quite a lot. We also do all kinds of workshops everywhere, share the sources, and then also make sure that everything we do somehow is open access, so that people can really read it. Because the worst thing I have is like when I'm reading a paper and it's blocked because we don't have all these things. I think the ImSwitch part I'm skipping a bit, but I'm inviting you to use this, or maybe to develop this. That's also co-developed with Jacopo. It's a very nice system. You go to ImSwitch, it's open source. Either use ours that's tightly connected to the Open UC2 system, or use the one that's officially published by the ImSwitch community. Yeah, and then I think that's pretty much it. So the one more last thing, but I don't want to show it because I don't have the electronics with me here. Forgot about it. What's also really nice is because I mean ImSwitch is nice, but you have to install Python. You have to be aware of Python in general. You can also use the microscopes in the browser. So that's something we have used recently. So there are two things. So Matchbox Scope is the one thing that I was showing earlier. So how to essentially build your espressoscope or Matchbox Scope step by step. What's also important is that you can connect to that over your browser. So you, for example, have this camera, you then connect to that over your USB serial. Right now, there's nothing connected, unfortunately, but this all works without installing anything. So just sharing the link also with chat. And what's also nice is so the other things. So if you want to control your microscope, we have this firmware. It's UC2 ESP. It's also open source. You can then flash your ESP with the firmware, connect the ports to your devices like motor and laser and stuff. And then when your device is connected, you can connect to the device and control it. And this works without installing any Python or any drivers. It's just working out of the box. And that's something I think which is really cool, because then it's really becoming reproducible, because I think everybody has a browser installed these days. And then that's really cool. Okay, I think this is pretty much it of this wild tour of different environments. So, and if you want to learn more, well, always feel invited to come to Jena, and also go to different websites that I've shown over the last few minutes, if not hours. Yeah, if you have any questions, I'm still here for a moment, but I'm also very happy to help you over Discord or so.
Great. Yeah, thank you so much, Benedict. There is one question that I see in the chat that hasn't been answered yet from Operov, who's asking, for surface evolution and droplet impact on fluid bath, they see the distortion of light due to interface. Can we use something similar for the shape of plankton?
For surface evolution and droplet impact on fluid bath. Okay, I'm not sure what fluid bath is. They see the distortion of light due to the interface. Okay, yeah. So I think this sounds a lot like holography, maybe. Because this is, I mean, if you detect the scattering, that's definitely possible. And you can also detect plankton, of course. So maybe just showing that this is also somewhere in the universe of the espressoscope. So if you go here and enter holo, maybe we're lucky, not 100% sure. Yeah, so this is one part. So you can acquire holograms with the the espressoscope. I think it's not super well documented, but yeah, here you see that maybe. So what you do is you take one image, and then you can virtually refocus. So this is computationally being refocused. So the first image is the autofocus, and then you see like at one point the filaments here, or the dust particles or whatever that is, become sharp, and you can literally refocus something that's not in focus. And so this is where holography is really powerful. But this is all in the manuscript. It has been uploaded to bioRxiv a few days ago, already last week on Thursday. But I think it's not available yet, so I can just quickly check. But there's more information on how, for example, you can use this really low-cost device on holography, and then also in particular for plankton, because many people use plankton for holography, or the other way around. Just checking. I think something is weird with my account or something. But anyway, look for espressoscope the next few days in bioRxiv, and I think there are some more information.
Great. Yeah, thank you. And Peter, do you want to unmute and ask your question?
Raman. Yes and no. So we measured some Raman. So the institute where I'm working at, the Leibniz Institute of Photonic Technologies, they are really big in Raman. But we simply used a commercial Raman spectrometer to identify what is it, the antibiotics, or like antibiotic resistances from bacteria. So we measured the spectrum of bacteria essentially, and found out that low-cost lenses don't have any problems, because usually what you have is autofluorescence also in glass. And since fluorescence is much stronger than the Raman signal, so Raman is well, nonlinear or higher harmonic or anti Stokes emission, then this is a lot weaker than the fluorescence. And usually you would see fluorescence from glass as well, but to our surprise, the low-cost lenses, they were completely fine. So we can do Raman even with low-cost equipment. Just the lasers and the spectrometer are quite expensive.
Great. And I also had a question just about the design process that you go through, because you built a lot of tools and some very frugal tools as well. So say you are trying to create a new part for the library for UC2. What is your thought process behind that? If you can kind of go through basic steps that you think about.
Yeah. That's a very good question. I'm thinking whether I have anything here on the table that makes sense to use, but I can maybe just give a quick walkthrough. Hopefully I'm partially prepared. Let's see. So let's imagine you have a lens with a certain diameter that you would like to put into the tube. Then of course you go online and know that there's a repository that's showing you all the. Okay, still screening. So where is it? Yeah, here. So template, I think it was called. And then you know the dimensions here. Here. Then you open whatever CAD program you have, and in best case, I'm connected and well prepared. So say we open a new device, sorry, a new part. So this is Autodesk Inventor. So this is then the full assembly for a more complex microscope. But then what you would do is you try to replicate the design. So what we've seen here. So what's important is it's 49.8 and 33.8 millimeters. So it's point symmetric. So what we do, we essentially draw a two-dimensional drawing. So I'm really bad at remembering stuff. So I mean, it should not. Oh, this is even in inches. Interesting. Okay, so it's 49.8 millimeter times 49.8 millimeters. And then what we also had was 33 point, what was it? Yeah. So we go 33.8 millimeters, and then all the way, and the same here. So this is a remote machine, so that's, oh. So all the way. And so this would already be almost sufficient. So we then can extrude the stuff here. But we also have these bars here. So then make sure that this has 45 degrees. And then make sure that they close. Same here on these edges. And then we have to make sure that they are having the right distance from the center, essentially. So I can show it to you. And then we are almost done. So we essentially just need to extrude the two-dimensional drawing. So this is it. And then, so we're almost there. So imagine you have a lens with a diameter of 20 millimeter, for example. Let's then add that to the drawing. So here, I think it was, let's quickly check. Was it 54 by 2? Like so. You can also reuse it so that it's more adaptive. So this is D9. But again, so you can use any CAD program like SolidWorks or Tinkercad or something like that. Okay, let's be accurate so that we could print it out and then actually use it with our system. So here again, and then the most important part, so D9, is then making the 2D to 3D conversion. Let's see whether this Autodesk is smart enough. So there we go. And let's say we want to make it, I don't know, maybe 5 millimeters thick because the lens is really thin. We just say it's 5 millimeter. And then we already have the insert part, which goes into the cube, like that here. And then if we want to add a lens, which is then being snapped into this thing, we can maybe be smart and say, okay, we really want to snap that in and make it like that. Make that here, and we cut it out. So then we say, okay, our lenses maybe, I don't know, I just come up with some random values. Has a certain curvature, and it has a certain thickness, and looks like this here. It's not really like so. And then obviously it has to, the light has to go through like this here. And since we want to snap it in, we have to maybe add some let's take the odds of inventor. Let's just make it smaller like so. And then this is almost it. So we just have to, I think, close the whole drawing and then make a revolution so that the drawing is actually, well, extrudable. And then let's hope that made it correctly. Not 100% sure. Let's see. That's good. Yeah. So, and then we have the axis of symmetry, which is this here. We subtract it, the Boolean operation. And well, I think we were not careful enough. So there's some very, very tiny section not being extruded. So let's do it like this here. And this would be essentially the design workflow for a very, very simple lens. So I'm not 100% sure whether this is actually working, but the hope would be that you press it inside and then it's sticking there, or you maybe glue it with hot glue, or maybe second part would be then to add a certain spacer that is then tightening it. So that's typically the design process, just very, very briefly. Hope that's
Great,
clear.
Thank you so much. Thank you so much, Benedict, also for sharing all of your previous projects and also ongoing projects as well. I know you sent in the links in the chat
Sure.
for everything that you were talking about. And so will you be available on Discord if people have more questions?
Yes, certainly. Yeah. So I try to so ping me, like write my name. I think it's Benedict, so maybe I just point that here for those who are still there. So if you add, I think Benedict or something, then I also see that. Sometimes I don't get notifications.
Okay.
Just name. Cool.
Great. Yeah. So then I think we can possibly end the your talk for the class, but we still have, I think everybody else needs to still stay online for a few minutes because we have some announcements to make for the course. But yeah, Benedict, thank you so much for hopping on. I know it's getting a little bit later there where you are, and we really, really appreciate it. Can we give him a round of applause?
Then have a nice or a good day then. Enjoy, and if you have questions, I would be very happy to help for all kinds of Frugal stuff.
Yes, of course. Thank you so much.
Thanks a lot for everything. Bye-bye.
Bye-bye. Okay, great. So everybody who is still online, we still have a few more announcements to make for the class. Maybe we can use this. Basically, we want to give you a kind of overview of what's going to be happening for the rest of the week. On Thursday, Manu will be back. He currently just landed in Senegal, but he'll be back on Thursday to talk a little bit about malaria. He is holding a workshop in Senegal on malaria microscopy and malaria diagnostics. So I think that'll be a fun discussion to have. And then on Friday, we are going to go through what Benedict just showed, which is how to use either AutoCAD or Tinkercad, any one of the programs, so that you can start to build 3D models of prototypes. And there will be two different options
for Friday's session of what you can build. One is the part for the UCT library. So Benedict showed how you would build a part if you want to fit a new lens to the microscope system. And then the other option is building a part for Inkwell, which is a blood smearing device that our lab has made. I can send a few links to that so you can take a look at that later. And we want students to choose which one they would be interested in building so that we can split you up into 2 groups and kind of facilitate a session where probably Rucha will be teaching one and then I could be teaching the other. And so it'll be a little bit easier to handle smaller groups. So the 2 options that we want you to think about, you don't have to decide right now, you can let us know on Friday, is a part for the UC2 library, which is the microscope system that Benedict just showed us, or a part for Inkwell, which is a blood smearing device that helps in malaria diagnosis. And I think final announcement is the active participation form. Do you want to talk about that, Rucha?
Sure. So we just want to make sure that people are like, drop deadline is happening, people are figuring out their schedule. So we will be sending out a form on Discord and over email, like probably today. Please fill it out by Friday. We just want to make sure we have an accurate and updated list of who is continuing with this class so that you're not left off on any communications or anything like that. So please check your Discord, check your email over the next day or 2 and have that submitted by Friday. For those of you in different time zones, it will be due Friday at Pacific time. So please feel free to convert that into your own time. But I think most of you are ahead of us. So if you submit it by Friday, your time, you will still be good. So yeah, just make sure to look for that and fill it out. And if you have any questions or concerns about what's on that form or anything like that, don't hesitate to reach out to us over Discord or email or whatever you prefer. And also just to get a understanding, if you're on Zoom, if you could do the raise hand, and then if you're in person, if you could raise your hand. Just curious how many people have done CAD, sort of, at least you could follow along with the tutorial that Benedict just did, or you've seen that before. There's no pressure. I'm just curious how many people are comfortable with that or not. Yes. Cool, good to know. All right, thanks. No one is expected to have a like strong background in it or anything. I just wanted to get a gauge of where people were. But that's exciting. So hopefully you'll learn something during lab. And then for the Thursday session, I guess this is more for you folks. I think we will plan to still be here and broadcast. If you want to come, I feel like this is nicer to see on screen, but if you want to join from your own laptops on Zoom, that's fine too. Does anyone like this better and say like want us to please be here to set this up specifically?
That will be only applying for the Thursday session. We will be available.
And it will still be remote on Thursday.
That was nice, but
Okay, yeah, I mean, I don't mind
Yeah,
coming, so,
we'll be here.
okay, we'll be here and set it up, but you're welcome to choose whatever mode you like better.
Yeah.
Also, sorry, just a few more announcements for the participants on Zoom. Is I know some people were curious if we could move the class to an earlier time. As of now, the official class will still start at 9:30 a.m. Pacific time, which is the same class time as usual. But we are debating whether we want to have a special 9 a.m. session, so 30 minutes earlier, just for global participants. If that is interesting to you, please let us know on Discord or through email, and we can try to arrange for that to happen. But right now, the regular normal class time is what we're going to stick with. And later on, we'll be working on projects very soon. So this class time, you don't have to stick with it. You can choose when you're meeting with your project teams. And we also had a few emails from students who are at Ashoka University who were asking if they could have a physical lab space to do their work in. We are working on that right now. Please hang tight. We'll give updates very soon. But for this Friday session, as long as you have access to Wi-Fi and a laptop, you should be able to do the lab.
If you happen to have a mouse that connects to your computer, like not a trackpad, please bring it on Friday. CAD is much easier when you have a real mouse, and not a trackpad. You can do it with a trackpad,
Mhm.
just in case you have it, it's way more convenient.
Okay, so tools for Friday session that you would need to get if you don't have or try to borrow or find a buddy who does have it is laptop, Wi-Fi, mouse. All right, if there are no more questions, I think we can end the session for today. Thank you so much all for joining, and we will see you on Thursday.
You-
Thanks for coming,
Yes. All right, thank you, everybody.